every organism, every body, human, animal, or even bacteria, is made of cells. One cell, many cells. We may have hundreds or thousands or 100,000 of cells. The cells have to operate. They have to perform the function of life. And the function of life is made by the components of each cell. Components that are performing the functions are called proteins, and they are responsible to perform all processes, constitutional and inducible. It means willing and unwillingly. Unwillingly means everything we do in order to exist, like breathing air, like digesting food. Willingly is what we want to do, and all has to be done by proteins. Proteins can function only when their structure, three-dimensional structure, is correct. This is what enables them to perform. And the structure is determined by the sequence. They are made of 20 amino acids that are bound together like a lace, like beads. A sequence of the beads is what is important, and this is dictated by the genetic code. The ribosome is the machine, the cellular machine, that can read the genetic code, that can understand it. The genetic code is made of 4 letters, and the ribosome understands which combination of 3 of the 4 is equivalent to introducing into the protein and the new amino acid. They know to read it, and they know to make the proteins from the amino acids. They are making them fast, correctly, and smoothly. There are many, many of them. They can reach millions in each cell, and they have to function constantly. These are the ribosomes, the machines, the small machines of the body. In the middle of last century, when it became evident that the DNA has the instructions of the components of life and proteins started to be revealed, the connection between the two of them was evident. However, it was yet not found. Even when it was found that there is a cellular machinery called the ribosome that translates this genetic information and that creates the proteins and that can understand both sides of the game, the reading and the creation, it was not clear how it can do it. Ribosomes work very fast. They make no mistakes, or almost no mistakes. They work under all types of conditions. They can make all types of proteins, those that are in the cell, they are soluble, or in the membrane, those that are working and those that are resting. It was clear that in order to understand how these functions are being performed, the structure of the ribosome is crucial. And to determine the structure of the ribosome, this seems to be a formidable task because of its complexity, its size, its instability, its tendency to deteriorate, and the fact that it can have many structures or conformations depending on the step in protein biosynthesis that they exist. Well, milestones. In the beginning, when we started to work towards the determination of the structure, it was clear that we have to have crystals. Otherwise, there is no way we can understand the fine, very detailed structure. This is correct for every molecule, for every cell component, for every salt, whatever you want. Crystals are needed. But how can one have crystals of something that is so complex, so unstable? People have tried to grow these crystals, obtain these crystals much before us. Actually, from the 50s of last century. We have started only in the 80s. Why did we think that we can get crystals when other people failed? Well, there was a story that was published at the end of the 70s about the bears of the North Pole. it was found that when these bears start hibernating in the beginning of the winter, their ribosomes organize themselves in a almost crystalline way on the membranes of the cells of these bears. Otherwise, I thought, when the bear gets up and has to function, the ribosomes will be completely deteriorated. The fact that they pack themselves and organize themselves seemed to be the way to keep the ribosomes from deterioration and have active ribosomes when it's needed. If the bear could do it, why should not be able to repeat it? So we tried to repeat what the bears did, but with better crystallization, better conditions, and in the test tube. And we thought that in this way, not only we will see how, if we determine the structure, we will see how the ribosome is built, but also how the active ribosome is built. So the first milestone was when we saw micro, very, very, very little crystals that gave the feeling or evidence that there is a potential in them, that if they can be improved, enlarged, and so on, they might be suitable for structure determination. It took some years until we got a bit larger crystals and some diffraction, which is required for structure determination. And this was the second milestone. Nobody believed this can happen. Actually, everybody, almost everybody, not everybody, all over the world, showed a lot of skepticism. But when we started to see some diffraction, we tried to improve that, and we found that crystals that grew after years and years of work were deteriorating while being measured, even in the first second. So we had to find a way to keep them alive, and we introduced a method that is called cryocrystallography, assuming that at cryotemperature the deterioration will be slower and we will be able to measure. And this worked and became popular all over the world within months. This was a very big milestone. I think from the technical point of view, the biggest milestone. While doing this and before having the structure, we tried to see how the ribosome is built in gross terms, and we used the electron microscope for this. And we found what was not believed at that time. The ribosome that is built of two subunits is reading the instructions on one side. This was actually known, but what was not known, it produces the protein that comes out of the other side through an internal tunnel. So here is the new protein and the tunnel that comes on the other side of the ribosome, which was not only not believed, but actually stayed controversial until 10 years ago, when somebody else could visualize it again by electron microscopy. So this was also a very big milestone at the middle of the 80s. I think the biggest milestone, of course, it was to discover or to determine the structure that you can see here and there. And even bigger than that was to find out how antibiotics bind to the ribosome, to find the mechanism how they stop the ribosome from working, to understand how resistance can be developed, and to understand how the antibiotics differentiate between the bacteria, the pathogen, and the patient.